Hello, everyone. My name is Rajeev Raghavan. I'm a senior biochemistry and molecular biology major from the University of Massachusetts Amherst. And today I'm going to be talking a bit about my research over the past and now concluding school year. I was interested in investigating the relationship between a missense mutation of the GABA3 gene in Donio by using a three-dimensional spatial motion tracking interface. So without further ado, let's begin. I want to start by talking a bit about some of the relevant concepts. First would be the GABA3 gene, which stands for gamma amino butyric acid type A receptor, and the 3 refers to the third subunit of the protein. GABA3 was originally discovered and studied in 1994, but at the time, most publications were focused on elucidating the function of the various subunits, studying its interaction with benzodiazepine ligands, or investigating its relationship with diseases like multiple sclerosis, depression, or schizophrenia, just to name a few. It wasn't until the mid to late 2000s that researchers started investigating the intersection between the GABA3 receptors and the central nervous system. The most up-to-date research suggests that rare missense GABA3 variants increase the subject's susceptibility to epileptic seizure disorders. Next, I want to talk about Donio. It's more commonly known as the zebrafish, and it's widely used in studies of the central nervous system and other neuroscientific studies. The zebrafish genome was sequenced in the early 2010s, and they have genetic analogs with over 80% of the polymorphisms associated with human diseases. Additionally, factors like being a vertebrate, having a large clutch size, and a quick larval development period makes them suitable model organisms. So I just wanted to mention the relevant research questions that these studies were intended to investigate or answer. The first one was, how can we track motion of multiple animals in a controlled setting? This question serves the scientific field beyond the scope of this specific project because it can be applied to other species as well. For example, observing nurturing or attachment behavior in a group of elephants, hunting patterns in lions, or whatever the case may be. If there's any correlation between locomotive behavior and some other psychological trait in animals, this would be a helpful endeavor. The second question is, what correlations exist between GABA3 mutation and susceptibility to epileptic seizures? In other words, what is the effect of the missense mutation on the GABA3 subunit? Is there something unique about a missense mutation at the alpha-3 subunit that induces epileptic seizures? And the last question that I was focusing on is, what specific behaviors will be observed in zebrafish that demonstrate epileptic seizure phenotypes? There are a number of characteristic behaviors that would indicate mutant phenotypes, such as sudden changes in direction, writhing of the fish, and C bends, which are when the fish's body contorts into the shape of a C, and an example picture shown at the bottom of the slide. Now on this slide, I've included a picture of the device that I created. It's not all that complex of a setup because all that was necessary was the ability to capture video footage of 2 different planar dimensions of a fish tank. The pieces include a Raspberry Pi 4 Model B computer with 2 cameras attached. One of the cameras is a Raspberry Pi 4 HQ 1080 pixel camera mounted on top of an Arducam tripod for stability, and the other one is a Logitech 1080 pixel C930E HD webcam. This was determined to produce slightly better image quality, so I used it for the front-facing view of the fish tank. The code I used ran the raspvid function with customization ability for parameters like the resolution, duration, bitrate, and frames per second. Once the video was captured, I used an SSH tunnel to send the video to my local computer and then used the VLC application developed by Videolan to convert it into a suitable MP4 format. Now let's move on to discussing the experimental setup. I have a sample video with the top facing or bird's eye view of the tank, and this was just a practice run with only 5 fish in the tank, so I'll go ahead and play it. Now, while this is playing, I'm going to talk about a few other things. So the volume of the tank was about 5.5 gallons, although a specific volume is not too significant in the grand scheme of the research question. I simply chose a volume like this to ensure adequate room for movement, but narrowing the scope to some extent. 10 fish were eventually used, but just for the purpose of a short example, this footage only contained 5. Each fish was shown at 96 hours post-fertilization and mutation. The cameras were set up about 3 feet away from the tank along each dimension, and each video was recorded for about 2.5 minutes with light tapping on the tank to trigger expression of the epileptic seizures. All experimental procedures and protocols were approved by the University of Massachusetts Amherst Institutional Animal Care and Use Committee. Data analysis was conducted using the ID Tracker program, which was developed at the De La Vieja lab in the Cajal Institute located in Madrid, Spain. The source code is available online for free. Overall, there was plenty of evidence to support the claim that this missense GABA3 mutant induces increased epileptic seizure phenotypes within the zebrafish. From a naked eye perspective, it was easy to notice some of the expected behaviors. Mutant fish often displayed erratic locomotive behavior coupled with swift changes of direction. Precise angular measurements have not been recorded yet, but I'm about to put a picture on the screen to illustrate a freeze frame of the analyzed video footage with ID Tracker. Although slightly difficult to discern, the picture shows clustering behavior along with movement vectors for each fish, which aids in the visualization of the swift direction changes. As we can see, the olive green and the red lines show sharp changes in direction. The takeaways for this are pretty wide-ranging. Not only has this process validated the quality of ID Tracker as it can track the movement of multiple erratic organisms in a small environment over a large period of time, but it can also generate movement maps that are three-dimensional for the whole recording process. Additionally, since GABA3 has a human analog gene, the findings from this and other studies support the linkage between GABA3 loss of function and epileptic seizures, a useful finding for clinical settings. Now, before wrapping up, I'd like to thank a few people. So first is Dr. Stephen D. Brewer, the professor of my seminar, and he helped me with developing this idea. The second person is Dr. Gerald B. Downs. I'd like to thank him for providing access to his laboratory and his zebrafish populations. The third group is the other members of the Open Science Instrumentation and Data Collection Seminar for helping with idea development and peer feedback. The fourth person is Xander Crowley of the biology department for managing orders of materials and electronics. And lastly, the Commonwealth Honors College for reviewing my proposal and providing funding for my project. Thank you for watching, everyone, and I hope you all enjoyed.